Chapter 4. Oh, I keep doing it. Chapter 4 is really all about intermolecular forces. So we're now taking what you learned in Chapter 3, all of the individual amino acids. We now jam them all together. Once they're now bound, what happens when they interact with the rest of the environment? Okay? Both with each other and with the solutions that they're in. Okay? Those types of interactions all come out of our intermolecular forces. So if you have any weaknesses with intermolecular forces, it is only going to get compounded in this section. Okay? So you need to be aware of the basic intermolecular forces, and we'll talk about a few extra ones that come out of this. Some of the bigger ones, you'll see the term hydrophilic, which meant water loving. What would we expect for intermolecular forces for a hydrophilic compound? We'd expect hydrogen bonding. You'll probably see some dipole-dipole. When we're looking at our amino acids, primarily hydrogen bonding is our big focus on. The other aspect for our hydrophilic is not so much, or not just hydrogen bonding, and not going down in polarity, but going up. Up in polarity puts us at ionic. Okay. So those two force classifications are very, very common in hydrogen bonding, or sorry, hydrophilic compounds.
And in chapter 4, when it was talking about the primary and secondary, it was talking about ionic bonds specifically, I believe. So we're going to go with that then? How that works?
Yes.
In formula structure.
Yeah. And you'll see a lot of how these forces, or they suggest that these forces affect how a structure flips and bonds and does all its thing. The strict complexity of it, we don't evaluate that much. Okay, really what we're talking about is, would I expect this amino acid to interact with water? Or would I expect it to not interact with water? Okay, and that's our primary focus. Which brings us to the other big force that typically shows up, would be hydrophobic.
Are you going to tell us if the molecule is hydrophilic or hydrophobic, or is it determined?
A little of both, but for the most part, you'll have to determine it. And this is why we talked about those forces initially in Chapter 2, because I want you to look at a structure with bonds and say what forces would possibly come out of it. So if you can recognize hydrogen bonding and ionic, what things would you need to look for within the structure? Well, ionic, you need to see charges. Hydrogen bonding, you need to see hydrogen bonded to nitrogen, oxygen, or fluorine. So if you see that in your bond structure, you know that's happening in your forces, which then means you're looking at a hydrophilic section. Okay. Hydrophobic is pretty much the opposite. It's water fearing, okay, or repelling. Repelling is probably more accurate for what it actually is. Ooh. Repelling?
Yeah.
Yeah.
Cool.
Hydrophilic is spelled wrong, but that's okay.
Oh, 2 L's. I try. Spelling doesn't count. And actually, seriously, it doesn't count on your exams. So when it comes to naming your amino acids, if you're close enough, I can figure it out. So if you spelled serine,
S-Y-R-N-E.
I'd give you a hard time.
Yeah, but the 3-letter is S-E-R, so.
Well, that doesn't mean you'd necessarily match it up, because some of them are close and some of them aren't. Okay, so I would probably, or even if you did something like that, I would probably accept it. If you spelled serine as cerium, I would probably have a problem with it. Okay, so there is some leeway in your spelling, but get as close as you can.
Histidine, because that's H-I-S-T-I instead of hista.
Oh,
You
yeah.
pronounce it histidine, but it's histidine.
And that one, if you threw in an A versus an I don't care. I'd circle it, say I, and that's about it. Okay. If I was feeling particularly cheeky, I might say minus 20 points and then not take off the 20 points. But don't stress about the spelling. If you got to spell it phonetically, You might actually write next to it phonetically spelled so that I know that that's what you're doing so I can pick it up.
On the single letter and the try to, because,
Yeah, not quite with
I write
those.
C, but phonetically I mean S.
Yeah, that's not going to work for the single letter or the three letter. Not a bad try, but the one letter and the three letter codes you're going to have nailed down perfectly. Hydrophobic, sort of back to our topic. Water fearing or repelling, what are we looking at for our forces there? London dispersion forces, so our LDF. So if we can recognize these, we can then start to say something about our our hydrophobic amino acids and our hydrophilic amino acids, which becomes really relevant when we move into tertiary structure, okay, which officially isn't on this exam. We'll talk about sort of how it shows up. Okay? So we need to have some understanding of those and remember back to them. And in once we move again into tertiary structure, what you need to be able to do is think about what makes proteins. Well, what does make proteins? The amino acids. Well, the amino acids can be classified according to hydrophilic and hydrophobic as two subcategories. Okay, so if you have an amino acid, say serine, right, and this is always a fun question that typically shows up in the more advanced classes, say serine, where do you find serine? Okay, is it in the hydrophilic range or the hydrophobic range? To answer that question, what do you have to know? You have to know the structure of serine. Okay? So I can get around having you memorize all of the amino acids, because what I can do is say, in the hydrophilic range, give me an example of an amino acid that fits in that range. As long as of your 5, you've kind of subdivided them over different categories or different functional groups, you can answer that question relatively easily. Okay? Taking it a step further, you will see amino acids show up on the exams. Okay? Well, if I just ask for an example of an amino acid found in those ranges, you can just give me the structure, or the one-letter code, or the three-letter code. So if you look through the rest of the exam and you find a structure of an amino acid that you would expect to be in the hydrophilic range, what could you do? Just draw it in that section. Okay? So take advantage of the whole exam. There can be answers given to you somewhere else that you can kind of needle your way back through to at least get partial credit, if not all credit. Questions? So, sorry, just a second. Go ahead, Alyssa. I heard you first.
For the amino acids that we memorize, can we just memorize like 5 in the same category, or is that going to be?
So this is where I was saying it's helpful to have them split across different categories. Okay. Minimally, one hydrophilic, one hydrophobic is going to be helpful. Most people will do that, and then you can pick the rest kind of wherever. I personally would prefer one in each category.
For us to memorize for the test?
For the test, yeah. This test isn't as big of a deal because we aren't looking at where the amino acids fall, okay, or what their properties are. For exam 2, we're looking at protein structure. Protein structure involves a lot more understanding of the hydrophilic, hydrophobic. You'll need some more classifications for that one. For this first exam, all I want you to do is know 5 amino acids, pick any 5 you want, and just deal with it. It's better if you pick them where you can use them. If you pick all 5 in one category, you can now only do answer questions in that one category and nothing else. So you've narrowed your field too much. For other tests, you should expand it out and cover all of them. Okay, all the primary categories, not all the amino acids.
I was thinking serine is hydrophilic then, right?
I should have picked one I knew better. Serine's with the alcohol, right?
Yeah.
Then yes, hydrophilic. Okay. Sorry, other questions on that? Okay. So chapter 4 kind of gets into the divisions of how we can look at an individual protein. So on that extreme left side of our image or our slide here, we've got that ATPase that we talked about before. What we see here is what's typically classified as the quaternary structure. Okay? So what's happening within our quaternary, our quaternary structure, which begins with Q and has some sequence of letters coming after it, we're looking at the combination of a bunch of different proteins. So in this structure, we have a bunch of different individual protein molecules that have all self-assembled around each other to build this enzyme. Okay, so our quaternary structure is the absolute kind of pinnacle of protein organization. We take a bunch of different protein individual units and we combine them into a larger structure, much like you look at the Power Rangers, maybe even Transformers to form the. Doesn't Transformers give you some big super?
No Power Rangers.
It's just Power Rangers? Okay. I can't remember. I thought Transformers did that too. Okay, so just everybody does it? They all combine and you have the individual units that you can then put together into one big massive awesome unit that then kills everything. Okay? So, except here it saves everything. Okay, so that's what's happening with our quaternary structure. We can then scale down and look at individual sections within that. So if we looked at this F1 section here, you'll notice that we have different colored sections. We've got a yellow and we've got a blue section. Each of those are colored because they are different proteins, okay, or different protein subunits. So what we have there is now an example of the tertiary structure. So if I look at just that blue, I'm now looking at the tertiary structure. So if I look at just one of those protein units, one of those peptides, how it folds and how it takes its shape is the tertiary structure. If I now combine that with another one, I can scale up to the quaternary structure. Right, but if we look, and that subunit's confusing as all heck, so we can look at the bottom section. And if we look at the bottom section, so this one was our, I believe this is the F1, yeah. If we look at the bottom section, that's our FO section. In our FO, you'll notice that they did do two coloring patterns as well. Okay? And if we look really carefully, they say B subunit and C subunits. Well, what's happening is a similar kind of situation. What we've got is different combinations coming down. So our B subunit is one protein that organizes with the C subunits. Okay? So that's, if we look at just the B subunit on its own, there's some tertiary structure. Okay? How does it fold? How does it then interact with the other subunit? That's moving to quaternary structure because we brought in a second protein. Within the C subunits, those are a little bit trickier because it turns out in the FO, it's not just one protein here. It's a combination of anywhere between 10 to 12 C subunits. So we can zoom out to this. This is our C subunit solely on its own. Okay? So we've pulled that one out all by itself. That, in its overall structure, is again our tertiary structure. It's how did the C subunit fold to look like that? When we put it into the FO, we have to combine it with 10 to 12 others. That combination takes us up to the quaternary structure again. So an individual protein on how it folds is our tertiary. If we look at our C subunit now at its sort of simplest level, okay, you may notice that there's some interesting things about it. Okay, one option is if we look way at the very top, you just kind of see a squiggly line. Okay? But if we look through the bulk of this, you notice that we have this kind of helical shape. And we've got multiple helical shapes. In fact, it's actually just two that are kind of coordinating near each other. Okay? Well, how those two helical shapes got near each other is an example of our tertiary structure. The helical shape itself is our secondary structure. Okay? So what we've got in this case, if I just look at this helical section, I now have secondary structure. So a portion of the properly folded protein, there are sections of it that form these particular patterns. If I recognize those patterns as showing up in other proteins, there's my secondary structure. Okay? How that secondary pattern interacts with another secondary pattern within the same protein gets us tertiary structure. How that protein with another protein gets us quaternary structure. How that sequence of proteins with another sequence of proteins, still quaternary structure. Okay? So we've got quaternary, tertiary, secondary. Anything else? Primary. The last one left over. Well, our primary is just the sequence of amino acids. There's no structure at all associated with it short of just formulas. Right, so we could look at the primary structure of that. One of the things that I like to reference in this case, if this is going to work, is we can go to the protein data bank. There it goes, maybe. Okay. So the protein data bank is a nice little source for a lot of biochemistry kind of people. And what we're looking at in this sequence, or this section here, is the C subunit, which, if we look really carefully, okay, right in the middle there, ATP synthase subunit C, it has 364 residues. Well, what's a residue? It's an amino acid. So to make the C subunit, I have 364 amino acids to make this single C subunit. Right? And if I want to know information about this, I can pull up the protein data bank where someone has actually isolated it and determined everything within it. And so you'll notice there's a lot of odd information kind of given to you there. The first thing in that green bar running across the top, they're telling you what it is. Okay, so maybe this sequence combines with something else and turns into a different subunit or folds differently. So in this case, they're just telling us that this is all part of the C subunit. The next sequence you'll notice is that we've got these kind of weird arrows and lines and loops and weird shapes. Okay? Those shapes are representative of the secondary structure. Okay? The next sequence, you'll notice we just have a bunch of seemingly random letters. What are those seemingly random letters? It's the one-letter code for our amino acids, which is our primary structure. Okay? So if I just want to know what amino acids are there, I can pull up Protein Data Bank, assuming someone's actually already solved it, and they can go through and they will give me the sequence of amino acids, which I can't highlight. Okay? And if we continue to go through this, we should list roughly 300 and whatever it was, 364 residues. Okay? So they'll list all of those out there for you. And you'll notice that we've got another little section here that's belonging to something else. Okay? So those are all of our amino acids. That's our primary structure. So what we'll do is talk about now the reverse. We'll work our way back up. So we'll start with the primary structure, then move to our secondary structure. That'll be it for today. Then we can talk about tertiary structure and quaternary structure next week. And then we'll talk about some enzyme stuff, which I'm sure everybody's going to love the enzyme kinetic stuff.
Is it the stuff from Gen Chem too?
It's worse, much, 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 much worse. And then we'll kind of move on, and I forget where we go after that. Yeah, I don't know why I'm forgetting where we go after that, but we'll move through. And lipids. We'll talk about lipid structures. Okay? So, back to our protein divisions. So what happens when we look at our protein primary structure? So this is ultimately just our sequence of amino acids. So we've, again, got our amino acids, and we've got our protein structure. I've noticed some people are still having problems with recognizing an amino acid and what's happening within it. So if we've got this amino acid sequence, you'll notice that they have highlighted some bonds in red. I'm not going to color over those in red because that would be kind of useless. And they're pointing to that first one, calling that a peptide bond. Very obnoxious as an organic chemist, because what they did was took a functional group that we all learned and loved in organic chemistry and gave it a new name. Why? I don't know. Okay. Someone, it's actually probably worse than that. It's probably someone that didn't understand organic chemistry and said, I'm not going to use anything organic chemistry uses, so I'm going to name it something else. Just to piss you off and me off. Okay? So that's now referenced as a peptide bond, which is ultimately just an amide bond. So if I want to look at the primary structure and I want to look at each of the amino acids that are located in it, the first thing that we'll see will be the nitrogen of our amino acid, because when we look at our primary structure, bless you, and all of our proteins, we always start with the amino end and we end with the carboxyl end. So it'll go nitrogen to our alpha carbon, where our R group is attached, to our carboxylic carbon with our carbonyl. There's amino acid 1. Amino acid 2.
So is that carboxylic carbon the beta carbon then?
Officially not in our labeling system. Our beta carbon actually comes off of the R group. Okay, and that's just how they went through and decided to name it. How do they reference the carboxylic carbon? What is that called? I'm going to need space to actually write this. It is the alpha carboxyl. Okay, because it is the carboxyl group bound to the alpha carbon. Okay, so our beta carbon is actually down on our R group, if there is a beta carbon. Okay. There may or may not be. If we have glycine, right, just hydrogens, there is no beta carbon. Okay, we can continue to go through this, and we can look at all of the individual amino acids that pop up in this, and we've got, what color have I not done? There we go, purple. We now have an amino acid that has a protein that has 1, 2, 3, 4 amino acids located within it. Okay. If we wanted to get fancy and use that whole peptide notation, we can call this probably a tetrapeptide, because there are 4 amino acids, and tetra is Greek or Latin for 4. Okay? So we would kind of think that primary structure is kind of meaningless. And in a sort of weird way, we can almost say that to be true, okay, within some exceptions. Where we've kind of classified primary structure as being important is that if I go through and change one of those amino acids, that now changes the primary structure, and that primary structure now affects the secondary structure. Well, if the change I've made in the primary structure affects the secondary structure, and the secondary structure changes. Well, if the secondary structure changes, tertiary changes. Well, if tertiary changes, quaternary changes. Okay? So we track that initial change all the way down to the primary structure, and that's kind of where we push the importance of primary structure, is that proper amino acid sequence. Even though it's really how the amino acid sequence is affecting everything else. Okay, so we kind of run into a gray area on where we would call that. But it's typically classified under our primary section. So why does the amino acid section or the sequence really matter all that much? So let's call this one that orange amino acid. Draw this in here so we don't have an R group. We got that hydrogen. We'll erase all that other junk so we can just focus on that orange. Okay, what amino acid was that? Glycine. Okay, so if it starts as glycine, what kind of intermolecular forces might we predict for this? Kind of go London dispersion, okay, which is not a bad direction to go with it, but I would argue it's a little bit different than that. I would almost argue a complete lack of forces. Why?
There's no hydrogen bonding, there's no ionic bond.
Not even hydrogen bonding, what did you suggest?
It's the difference between carbon and hydrogen, which is nil.
This hydrogen is tiny, which means if we look at the rest of the structure around it, it's buried kind of inside. That hydrogen isn't going to see the light of day. Okay? It is so tiny that it's just kind of meaningless. So if I look at glycine, I would kind of classify glycine as almost as a lack of interaction. Okay? Definitely cool with the London dispersion. That is absolutely strictly correct. But in this case, it's so tiny, it's kind of buried back in the chain. Very unlikely for it to actually see or do much of anything.
Are you only paying attention to the R group?
I'm only looking at the R group. Why not look at the backbone?
Because they're all.
So there's 2 reasons. They're all
Interacting with one another, so there's no charge whatsoever.
they're all bound into the backbone, which means I don't care about that. Okay? And in particular, when we're looking at primary structure, we're really just focused on the R groups. But the backbone we don't care about because it's in the backbone. It's the same all the way across, okay, for our primary structure. The only case where it would be different is at the beginning and at the end. And in a short peptide like this, that's a pretty large contribution. But in the C subunit that we just looked at, which is actually a relatively small protein, there were 364 amino acids. What percentage does the beginning and the end contribute to 364? Virtually nothing. Okay, so percentage-wise, the amine and the carboxyl end are relatively minimal in our overall structure contribution. They are still there, they are still important. We have to evaluate them when looking at charge. But as far as understanding what's happening with our primary structure, I don't care.
Because it's really only the R groups that are going to interact with other proteins or interact with itself, fold on itself, or interact with other.
Yes and no. Yeah, that's usually how I would interpret it. It's a little bit off when we look at secondary structure. Secondary structure is actually based off of the backbone. Okay. So glycine, kind of back to our story, relatively no interactions. So what happens if somehow in our translation or transcription process, that amino acid is converted to, hopefully I did the structure right. That's what I'm trying to draw. I'm not sure if I'm right. It should be tyrosine. I think there's an extra carbon in there. So if we get a mutation where we now have tyrosine, okay, it should be darn close at least. Okay. So then the question is, should that change anything? Okay. There's a couple big reasons why it should change. Our strongest intermolecular force coming off of tyrosine is hydrogen bonding. It's such a strong force that we actually have a third pKa for tyrosine. Okay, so we started with an amino acid that had just barely London dispersion forces and in all likelihood no interactions. And we've now switched it to an amino acid that actually has a third pKa, which means that can potentially take on a charge. Is that charge going to have a large contribution to the overall structure? Yeah, that can have a massive effect. Take it a step further. We started with hydrogen with virtually no interactions, and we now switched it to carbon and a massive ring. Maybe that glycine, when it folded up, was bound in a tight little pocket. Okay? And since it's tiny, it can sit in that tight little pocket. But if we now have tyrosine, can it sit in that tight little pocket? No, so the protein necessarily is going to have to open up to allow that tyrosine R group some space. Okay, well, if it opens up, that affects potentially the secondary structure, for sure the tertiary structure, which then could affect quaternary structure if it needed to get up to it. Okay, so a single point mutation within an amino acid sequence can have a massive change in the overall structure. To figure that out, you have to know what your amino acids look like, which goes back to why in a regular class you're supposed to memorize all 20. Because what the question would be is, this was glycine, it's changed to tyrosine. Is there an effect? If you have no idea what the amino acids are, you can't answer that question. Because there are some mutations that won't have a major effect. For instance, if instead of it being tyrosine, I switched it to which amino acid is this? Alanine. Intermolecular forces, London dispersion. It is definitely bigger than a hydrogen, but it's still relatively small. Would I expect a mutation from glycine to alanine have much of an effect? No. So I'd have to know something about those structures to be able to predict how this is going to affect the overall structure through the rest of the molecule, through the rest of the peptide.
So you're not just looking at the size of the R group, or you're actually looking at the bonds in the R group with the size?
Size contributes and functionality or classifications. Okay. So yeah, everything stacks.
What would be an example of one that there were different bonds, but they're relatively the same size, and therefore there wasn't a change in...
Glycine and alanine. The difference between glycine and alanine is almost non-existent. Methyl is still bigger than a hydrogen, but not by much. Okay. So we would expect potentially some size effects, but wherever the glycine was, an alanine with an extra methyl probably isn't going to change the structure that much. Okay. So what you could go through and do is with your amino acids, look at that table and say, okay, if I had this one, what could I change it to have no effect on the overall structure? Okay. What could I change it to that would have a drastic effect? Having some understanding of those structures and how they can affect the rest of the molecule is really what primary structure is about. It's looking at that. Okay, and so one of the questions that we could potentially run into is, really, is it all that big of a deal? Well, we just discussed how it can be a big deal theoretically. So there's your structure examples. You should know all your structures. But in actuality, it's very important. Right? What we've got here are normal red blood cells and sickled red blood cells. They've gone through it and looked at the protein codes, and they've found that it does have a difference. That one difference happens to be at amino acid 6. You switch from it's glutamate or glutamic acid. Okay, glutamine is the GLN to valine or valine. Okay. What would we expect that to be a difference? Let's see, did I give the structures here? Well, so we look up the structures. Glutamate to valine. Would there be a significant change in the overall structure if I do that swap?
Yes.
Yeah, I got something that is charged as a side group at neutral pH to something that is now uncharged. Okay? That is a significant change in the overall structure. And we move into, say, tertiary structure, we can see how big of a difference that really makes. Because we could also evaluate size. I would probably argue in this case we don't have that much of a size difference. We still have this kind of Y shape. We're missing some of this, but we got this to replace it. So in kind of a very rough approximation, you're really just saying charge. Okay. And that single point change takes a healthy blood cell and turns it into a sickled blood cell. And sickled blood cells gives you sickle cell anemia, you can't transport oxygen as well, and you get all sorts of health side effects to come along with it. So one change can make a massive difference. Okay. One of the things that's also kind of nice is one change could also potentially make no difference. Okay, which is kind of one of the things that is fascinating and useful about life. We can have some mutations, and those mutations can still result in the cell living. There is also a possibility that it can result in it dying, okay, depending on the level of the mutation, okay, and where that mutation occurred. So looking at your primary structure is important. You have to be able to evaluate and look at those amino acid structures and say, okay, is this an important relationship or is it not? How would I ask this on the test? Anybody have the practice exam printed? Anybody have it printed?
Yeah, right here.
Yours looks closest.
Are you looking at that one?
Question D. In attempting to determine which residues are essential for proper protein function, the primary structure is often altered by substituting different amino acids. Circle the amino acid that would least impact protein folding after substitution in the primary structure. So the original amino acid is given, and if you look at the question D, you've got 5 other amino acids drawn below. Right? It's an interesting question because it gets back at this point. What's even more interesting about it is you can argue there are a lot of different answers. Okay? That's why you need to explain your answers. Okay? So you would look at that structure and come up with the best explanation for it on what you see to be the best relationships. Okay? So that is a good and simple, relatively simple question. I don't have to have you memorize all of the amino acids because I can give you all the structures. And you can interpret off of those structures. Yes?
So I have a question. So as we go through it, we're looking for the one that would give us the least amount of bonding alternatives. It wouldn't fold back on itself, right?
We're looking for the one that is a closest match to the original amino acid.
Right.
Okay? And you'll notice there aren't a whole lot of really close matches within that. So this is where you would have to decide by looking at that and saying which of these matches best, which of these would have the least change in intermolecular forces in that overall process.
So with that,
So in this case, we're comparing, so I believe it's serine, right? Serine, and you're now saying, switching it out to glycine. Well, serine has that one carbon and an OH.
right.
Glycine has the one carbon. As far as size goes, is there much of a difference? No. So according to the size argument, yeah, I would completely agree with you. I think that makes sense.
But you're missing the OH group.
But you're missing the OH group. And glycine just being a methyl gives us London dispersion forces and
Right.
London dispersion forces only. OH gives us hydrogen bonding. So based on size, yeah, your argument makes sense.
Okay.
Based on intermolecular forces, you run into a slight issue there.
Okay.
Right? Which one trumps? That's actually what I'm getting at with the question. I want you to come up with the best explanation for you. And as long as you can convince me, I'm fine.
Okay.
And there are multiple answers in there that would convince me. Okay? Other questions? Yeah.
So in answering such a question, you have to consider the size, the charge, the similarity, and all
Size, charge, intermolecular forces, are all of those things important to how that molecule interacts with other molecules? Yeah. So should you evaluate those things?
How should
Yeah.
we look at size? Is there like a discrepancy, like if you count like the carbon chain, or just like look at the
Is
shape and then?
one carbon a different size than two carbons?
Relatively?
Very slightly.
No.
So slightly. Is one carbon different than 6 carbons?
Yeah.
Okay. So it's not a strict science. We're saying we're taking our best guess at what's happening, and you just need to defend your answer. Right? If I look at it and say you didn't consider 4 out of 5 possible things to evaluate, and I go, that's kind of questionable.
Gotcha.
Right? The two primary ones you would look at would be size and intermolecular forces, because those are our largest contributors to all chemistry. Other questions? Okay. Secondary structure. So when we move up to secondary structure, this is where it gets kind of interesting in my opinion, because we actually now kind of ignore our R groups. The side chains become less important. And what starts happening is we're looking at how the backbone of our amino acid interacts with other parts of the backbone. Okay, so we end up doing different rotations, and that's what they're trying to show in this figure, about these two bond angles. Our psi and phi? Do we remember? Is it phi?
Yeah, fine.
Psi and phi. Okay? It's those two angles, or those two rotations, that can now change how our molecule interacts. If it is drawn in this fashion, how does that show a potential interaction? Well, if it's in this orientation, this oxygen is doubly bonded to that carbon with 2 lone pairs. What can that oxygen do? I don't like the word bond. It can accept what? Hydrogen. It can donate what?
Electrons.
Electrons. Okay. So this could be a hydrogen bond acceptor. Okay. Well, if we look below in that structure, we have a hydrogen bound to a nitrogen. Nitrogen being more electronegative than hydrogen, that hydrogen becomes partially positive. With that oxygen being partially negative, what can potentially occur between those? Hydrogen bonding. Okay. We have, and this is kind of bizarre, we have an intramolecular force, which is really, really weird. Okay, because if we think back to that previous slide where we look at all the interactions potential or potentially possible, that's really based in organic chemistry where we simplified things down. We look at intramolecular attractions being a bond between those two hydrogens. And we look at intermolecular attractions, the interaction between two separate molecules. In biochem, our structures aren't these tiny little simple organic molecules. They are now these massive, massive structures. Well, what can a massive structure do? It can fold on itself and interact with itself. So we can get an intra, so within the molecule, attractive force, not a bond. Okay? It's that intramolecular force that can now cause different structural features to occur. Okay? And that all really depends on the rotation about psi or phi. Okay? What are those exact rotations? I think the textbook starts to talk about them. Ultimately, that's just a sequence of numbers. Okay? I don't care about the sequence of numbers. What I care about is, do you understand that you can get that intramolecular attraction? Okay? Well, can you? Yeah. Okay? Where is it going to occur? Okay? Would I ever expect it to occur between this hydrogen and this hydrogen? Why not? Okay.
They're both partially positive.
The biggest answer is that they're both partially positive. Okay, one possible answer you might say, well, they aren't close to each other. Can I change that? They can be, because I can rotate about those psi and phi to get them close enough to each other where they can potentially interact. Okay? So it's evaluating those intermolecular forces and saying, what can happen here? How does this affect my overall structure? Okay? So our forces, we're primarily looking at hydrogen bonding. Those bond angles are referred to as our psi and phi. They're called the Ramachandran angles, named after the guy that first found them. Okay, I believe that's what happened with that. Okay. Recognizing that they exist is important, so I might ask with an individual amino acid, what bonds are you concerned about rotating about? Okay, well, within an individual amino acid, there's really only 2 bonds that you'd be concerned about rotating in secondary structure. What are those 2 bonds? So if we draw a simple amino acid, a simple amino acid, and this was going to go into a peptide chain, yes, our structure would change. There's our R group. There's a lot of bonds that we could potentially look at, but when it comes to secondary structure, where do you need to be looking? You're looking at this bond angle, or not angle, but that rotation, and you're looking at this rotation. Do you need to know which one's psi or phi? No. Right? Do you need to acknowledge that those are the bonds that you're rotating? Yes. Okay? Whoa, that jumped way faster than I wanted. There are a lot of examples of protein secondary structure. Okay? Our backbone interactions, so it's how does the individual backbone, which is why you're seeing the ribbon here, interact. If it interacts over short distances, okay, like we were kind of alluding to with that previous example, those short distances are going to result primarily in alpha helices, because we're looking at the backbone interacting somewhere along it relatively close to it. If we're looking at an interaction between a peptide or a protein sequence that is a long distance away, well, that long distance isn't really going to be an alpha helix, because in an alpha helix, we're taking that chain and we have to interact all the way around. So we get very local interactions. In the beta sheet, okay, or our long distance interaction, we take our peptide chain and it folds back on itself. Well, if I have my peptide chain and these 2 parts are what are going to interact, are those close to each other or far away from each other? Relatively far from each other, okay, to form your beta sheets. So we're looking at this kind of short versus long distance interactions. And by short and long, we're not saying the distance between the atoms interacting. Okay, what we're saying is the distance in the primary structure between the atoms that are interacting. Because in the primary structure, it's simply the chain. Okay, in our secondary, we can take amino acid 55 and interact that, 55, with amino acid 463. Okay, that is a relatively long distance away in the primary structure. In the interaction, it still has to be very, very close. That's why it's an interaction. Okay, so when we're referring to short and long distances, that's what we're talking about. It's relative in the primary chain. Within these, okay, it would have been nice if I'd actually kind of stepwise it outwards. We've got our alpha helix in this big, nasty, ugly-looking structure. And we've got our beta sheets in this other big, nasty, ugly-looking structure. If we compare those 2 middle structures to each other, is it obvious the difference between them? Just look in the middle. That may be kind of difficult for most people to see. Okay, I know I would have a hard time seeing it. So let's move stepwise outwards. What's happening as we move outwards in both cases? What have we simplified? We removed the R groups, and we're only looking at the backbone. Okay, in both cases. Why? In secondary structure, do we care about the R groups? No. That's not our primary interaction. We're looking at the hydrogen bonding of the backbone, not the R groups. So we can simplify our entire molecule into just looking at the backbone. Now when we look at the backbone, it makes it a little bit cleaner to see. Okay? In the second example with the beta sheet, they made it a little bit easier because what did they overlay over the top of the structure? These sort of beautiful colored yellow lines. What are those yellow lines supposed to represent? The backbone, and in this case we have 3 strands of the backbone. When we take a look at, where did it go? Our alpha helix, where's our backbone? How many backbones are there? There's only 1, followed through that green bond. Okay, not as easy to see without the ribbon, but if we drop the ribbon, drop a lot more of the backbone structure, and just put in now an occasional atom, we see this nice ribbon structure. We get a single strand in our alpha helix. In the beta sheet, we're looking at multiple strands. Those multiple strands can be part of the same protein. It's also possible they're part of different proteins. Okay. Since we're looking at secondary structure, which of those 2 options is the most likely or the better answer? Are we looking at the same protein or different proteins forming the beta sheets? For secondary structure. What we're comparing is just the words. They aren't up there. We've got this beta sheet, or this sequence. We've got these strands all aligned with each other. One option is those strands all belong to the exact same protein. The other option is that some of those strands belong to protein A, some of those belong to protein B. If we are looking at secondary structure, which of those 2 options is correct? Same protein or different proteins?
Same.
Must be the same. Why? Secondary structure. Multiple proteins does not show up until we get to quaternary structure. Okay. So since we're in secondary structure, we can make the assumption that these 3 strands are all part of the exact same protein. Okay. With our alpha helix, it's a little bit easier. There is only 1 strand. Okay. And it's not saying that this is the sole structure of our protein. Our protein can continue off and do other stuff somewhere else. But one section of it gives us this helix, gives us this ribbon-like shape. Because one section of it has that, we refer to that as a motif, because that motif shows up in multiple different proteins. Same thing with our beta sheets. Okay, I keep calling them by their names, but our ribbons that are kind of overlaying with each other. It's the same general idea. We have the option of having our protein then continue off some other direction and folding back to this. What do those arrows mean?
Directionality.
It's the directionality. Well, where do we start our amino acid? The amine. So what's hanging out over here? Our amine group.
Is that what they were talking about when they did the zigzag structure? Is it zigzagging back?
Because if we turn it sideways, yeah. So our sheet runs up, does something, who knows what, comes back down. Then does something, who knows what, comes back up. And if we said this was the end of our amino acid, what would be out there? A carboxylic acid. Does it necessarily have to follow where this middle section is the middle of the protein? No, not necessarily. Okay, it just kind of depends on the protein itself and everything else. We don't know necessarily what's happening out in those ranges. And we don't ultimately care when it comes to secondary structure. All that we care about is, do we see this sequence where we have the proteins aligned with each other, the backbones aligned? If we have several strands of the protein aligned, we refer to it as a beta sheet. Okay. If we happen to see our protein align and give us this kind of helical shape, then we have an alpha helix. Those are our 2 primary forms of secondary structure. There's all sorts of other characteristics that can be associated with those. What are the proper psi and phi bond angles associated with that? Okay? You can look at how many individual amino acids occur per turn. Okay? What amino acids or what part of your backbone is interacting with the other part of the backbone? Those details are listed in the textbook. You do have questions on them in the homework. Are they super relevant to what we need to get out of this class and what you'd be tested on? No. Okay? So some of the homework problems are there as an exposure, right? Have you seen it, at least worked with it, attempted it? Now prove that you understand the overall concept. That's what I want to see in the exam and in the class. Okay? Questions? So, oh cool, I do have the video linked. We got another video today. Is that going to do it? Yes. So this is, oh, not the video I thought it was. So what they've got here is another alpha helix. And what they're trying to do is look at the individual side chain and explain what's happening, or the backbone of it, and look at all the individual pieces and look at the orientations of how they match up, where those individual R groups are aimed, where the backbones, hydrogen bonding, all of that fun stuff. Can I pause right there? So you'll notice that our R groups are aimed in all cases away from the helix. Well, should they be aimed away from the helix? Yeah, because they're aimed inwards. How do we helicate? Helicize? If we take a look at our backbone, they're highlighting the nitrogen and the carbonyl. Okay? That's our nitrogen from our first amino acid, wherever we start within our backbone. Okay? It doesn't matter, again, doesn't matter where, we just pick an amino group. That is now going to hydrogen bond with the carbonyl that is 4 amino acids further away. Okay? Why does it have to be 4?
It's forming a helix.
It's forming a helix. Could it be the second one? Okay? Well, if we've got these amino acids that are then kind of twisting away from each other, if it necessarily twists, the amine's going to be over here, or carbonyl is going to be over here. So right away, they can't orient. So if we I'm going to aim back the other direction. As we continue to twist around, eventually we'll align where they're on top of each other. That stack can get us that interaction. Okay? What if we changed the coil of the helix? Okay, made it a broader coil. Would that change the interaction here? Yeah, necessarily. Could we make it tighter? I don't think we can, actually. I think this is as tight as we can really get it. Okay, but we could potentially make it a broader helix, and we're going to get different amino acids linking up. Okay, the most common one happens to be set up like this, where your first amino acid links up to now the 5th amino acid in the sequence. Okay, and I think there's audio with it potentially too, where they're talking about the hydrogen bonding. You'll notice that in our drawings it's very difficult to see that three-dimensional structure, which is why they try and rotate this and spin it and slowly kind of work through it. So you can see those individual rotations and those motions. This is a fun view from the top. You can see that helix straight down the middle. Okay. So now they go through and label in all your hydrogen bonding interactions so you can get some kind of cool video of those three-dimensional images, okay, when it comes to those helices. So it's kind of neat. That's where the ribbon comes from. They do that to simplify the overall structure so that we can see that this pattern does exist. Now that we know it exists, we can simplify our structure and not have to draw all those side chains. Okay, thank you, those people. So, what conditions might prevent our alpha helix from forming? Interesting idea.
The lack of hydrogen bonding.
Are we going to have a lack of hydrogen bonding through the backbone?
No.
No, it doesn't matter what amino acid we have, we're still going to have a nitrogen and a carbonyl.
Charge of the amino acid or the carboxylic acid?
What do you mean? That's not in the backbone.
That would be the pH, wouldn't it?
So pH can affect this potentially, but it's going to come back to structural effects. And the amino acid, you actually have the right idea, but that's not in the backbone. Why would that have an effect?
Bonding to in the middle.
It has nothing to do with the arc, it's going to be pointing away from the alpha helix.
They're pointing away from the alpha helix. So let's say we pick those two. There's one aimed that way. The other one, shoot, already lost my structure, aimed that way. What if those two groups are massive?
Sterically hindered.
They sterically butt up against each other, and what are they going to do?
Rotate.
Rotate completely away from each other. Are we in the alpha helix now?
No.
No, we have to have that appropriate bond angle. So if we have side chains that interact in a bad fashion with each other, where they need to get away from each other, that's going to prevent the alpha helix. Okay? Is there another way we could look at it? Is there, say, an amino acid that changes our ability to rotate about those bonds?
Wouldn't we only be speaking of deformities in this case? Because when we're forming these amino acids to form these proteins, it's going to want to fold the alpha helix. And if we don't have
No,
alpha helix,
not true. If I take a protein that only wants beta sheets, does it want to form the alpha helix? No, it only forms beta sheets. So if a protein wants to be an alpha helix, it's going to set up as an alpha helix. But the amino acid sequence must be appropriate that allows for the alpha helix to form. Otherwise, that protein doesn't exist. You're looking at a chicken-egg argument. Okay, what I'm saying is if we have an alpha helix, okay, or if we're trying to look at an amino acid sequence and say, can I have an alpha helix? Well, if I have two amino acids that have massive groups that need to get away from each other, I cannot form an alpha helix there. So what we're trying to decide, is there something about looking at the sequence where we could say, that is alpha helix possible, or that is alpha helix impossible.
Okay.
Right? So it's impossible if our R groups butt up against each other. Right? Is there potentially another amino acid that prevents the rotation about that psi or phi angle? What's that? Large is going back to that steric argument, and that's not going to prevent the rotation as much.
So we're talking about secondary, right? Secondary structure?
This is still part of secondary structure.
I
So
thought
the
you said
secondary
ours didn't
structure,
matter, so that's why
they
I'm-
don't matter to your primary or your, I can't say primary, to the actual helix. So we kind of ignore that. But the R groups can prevent the secondary structure from occurring.
Okay.
Okay? And one of those ways they prevent is if there's steric hindrance, steric blocking that prevents the proper orientation. Is there a functional or an amino acid that does not allow for rotation about our Ramachandran angles?
Is it when you have two cysteine atoms?
So we're looking for a specific amino acid. Those of you with a book or have the amino acids out in front of you might help. Flip back. Actually, we can cheat and go all the way back. Oh, I'm going to regret that. If we take a look at lysine, where's lysine on this chart? How do they organize it?
Third one down left.
Third one? Oh, that's leucine.
Third from the bottom on the right.
Oh, yeah. Lysine. Does that prevent rotation about those two bond angles?
Yeah.
No. Those can freely rotate. That's not a problem. You want a big hint?
It's tryptophan.
Not tryptophan. What was that? Take a look at proline. Where are bond angles?
Or within the ring.
We have a bond angle there. That's free to rotate, but can I rotate about the carbon-nitrogen? No, it's locked in with the ring. Proline can mess with our ability to align those proper angles. Okay? So proline is not likely to be found within an alpha helix because it's going to prevent that proper rotation.
Could you say that with really anything with a ring, though?
Anything with a ring that includes the backbone of your amino acid. How many amino acids include the backbone in their R group? One. Proline.
Yeah, that works.
So yes, you're right, technically.
Say that again? Sorry.
Anything that includes an R group bound to the backbone of your amino acid would prevent this proper rotation. Okay, proline's the only one that does that. Okay. What if we had a sequence of a bunch of, where are they? A bunch of, say, glutamates, or glutamic acid. Glutamic acid, glutamic acid, glutamic acid, glutamic acid. Would that potentially cause a problem with our alpha helix?
It could.
Why?
If you have another carboxylic
Why?
acid group, well, it can hydrogen bond with either or.
So we can't I mean, each of those could hydrogen bond with water, but what happens when those negatives get near each other? They repel. So instead of having a steric interference, we actually have electron clouds interfering with each other, actual charges, which could potentially be problematic as well. So within our alpha helix, we want relatively small groups that don't interact very much with the next one. Okay? If we got big, bulky steric groups, he's really excited. Okay? That's not going to allow for the proper stacking all the way through it. Okay? So we got a bunch of random facts. They're typically clockwise right-handed turns. Any ideas why? Has to do with the chirality of your amino acids. Tends to force this clockwise rotation. You get 3.6 amino acids, so here's that big list of numbers on what things happen within this. All this fun stuff that we can go through and look at. I'm not sure that I really want to talk about those other than we just have a bunch of facts associated with our alpha helices. So much for doing a review. Our beta sheets. Okay. In the beta sheet shown, we've got one going up, one going down, one going up. Do they have to alternate? No. I could have it where they all go in that up direction. How does that work? Well, the first one goes up, does a bunch of junk, 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 up again. Junk, 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 up again. Okay? So we have both of those options. Because both of those options are possible to us, we end up calling them by different names. This is where we get parallel versus it is anti-parallel, yes. Yeah. Parallel versus anti-parallel. Okay? Parallel, they're all, your beta sheet direction is amine to carboxyl, amine to carboxyl, amine to carboxyl. The anti-parallel is amine to carboxyl, amine to carboxyl. Okay, so it's snakes as opposed to just all the same alignment. Okay? Our R groups. What's going to necessarily happen with our R groups? Well, we take a look at the image. I know it's not the easiest to see. Where are your R groups aligned? These are aimed. Careful. These R groups are aimed towards you. These R groups are aimed away from you. So what's happening if we look at our beta sheets as my arms, okay, is our R groups are aimed alternating up and down. Or sorry, up, and then the next one would be down. Okay? And depending on which chain we've got, our R groups are always going to stagger away from the beta sheet itself. Okay? The hydrogen bonding that's occurring, I know it's not super easy to see, but what you're looking at is, again, that same hydrogen bonding interaction between the carboxyl carbonyl and the nitrogen NH, which is kind of showing up down here. You can see on the edge here that they're showing those red dotted lines. That's where they're trying to show the interaction, where it would show up with the next chain. Okay. Again, with the beta sheets, you can come up with all sorts of facts and numbers and all that fun stuff. Again, I don't particularly care. I'm more focused on what is a beta sheet. Well, it's a secondary structure. We have two possibilities, parallel or anti-parallel. Okay. Slightly cleaner drawing of how this aligns. You can go through and now see where your hydrogen bonding interactions occur. You'll notice that we run in that uppercase, we're doing which direction? Parallel or anti-parallel? Parallel. We're both running nitrogen to our carboxyl from right to left. In the bottom case, we're looking at the anti-parallel. Okay, you'll notice that, again, we also get slightly different hydrogen bonding interactions. The anti-parallel almost lines up perfectly. The parallel, we've got some kind of staggered interactions with it. Would that change how our molecule could potentially interact with other things? Yeah. Okay. Do we need to know that? Not really. We just need to know that we're looking at different sequences. If I provided these two structures, see, can we do it with this? No, I can't do it with this. Never mind. I don't ask that question. Okay. So we've got our beta sheets. We're again looking at the hydrogen bonding and not backbone. U-turns. Oh, that's a nice figure to put there. Your textbook talks about it a little bit. What we're really referencing is if we take a look at our anti-parallel at the bottom, we're running here as our direction goes, but then what has to happen? Eventually I have to do what? I have to turn around at some point. When and how I turn around is kind of the definition of our beta turns. Okay? If we turn around immediately, okay, or relatively immediately, that's where we get our beta U-turn. What amino acids might we expect in that beta U-turn?
The one we just talked about, like the
Proline's a good option. Why? Because proline's going to force that bond angle to jam backwards on itself really fast. Okay, what else might we expect to see in there? How much space are you seeing in these U-turn positions? Not much at all. So what amino acids might we find there? Glycine, alanine, the tiny ones. Would we expect to see big ones there? Probably not, because they'll start to sterically bump into each other, which is going to prevent it from folding back on itself. Okay? I know for a fact that there was a question that went into a lot more detail with the beta U-turns and sapling. I'm pretty sure I deleted it, because I went back into the textbook and there was no information there, and I have no desire to talk about beta U-turns any more than that. Okay? So, questions? Last thing we'll kind of hit on is what the textbook refers to as secondary plus. Okay, well, our next step from secondary is our tertiary. And our tertiary is how, now that we've got these secondary sequences, how do those secondary sequences interact with the rest of the protein to give us our tertiary structure? Okay, well, if we take a look at these, and this is where it gets a bit weird, we've got a beta sheet. Right? In our standard beta sheet, we just see a sheet. But what happens if that sheet folds back on itself? Comes a barrel. Right? So take a sheet, there's our beta sheet, and it folds back on itself. We now have a barrel. Okay? Is that secondary structure or tertiary structure?
Still secondary.
Okay? That's where it gets really weird. Our secondary structure is just the structure itself, just our sheet, interacting with no other aspects of our structure. When we now take this beta sheet and we interact it with another beta sheet, oh, now we have tertiary structure. We have these two separate beta sheets. But what happens when I have a beta sheet that interacts with itself? Well, that's kind of weird, because that's not really interacting with another beta sheet, and it's not really it's not really the same beta sheet. Okay? So you're getting a beta sheet interacting with itself. That puts you in this kind of gray area between secondary structure and tertiary structure. Okay? Your secondary structure is just supposed to be the sheet itself interacting with nothing else. A beta barrel is that beta sheet interacting with itself. Okay? Why do we call that secondary? Well, it's because it's interacting with itself. If it interacts with something else, then it's really tertiary. Okay? And this is where your definition of these primary, secondary, tertiaries really does fall apart. Okay? And they call these as motifs. If we look within an individual protein and I see a section, say like a beta barrel, and I look at another protein and I see something just like it, but there's still a bunch of other junk in the protein. Oh, well, that's a common pattern that I can see in all proteins, just like we did with alpha helices and beta sheets. If I look at all proteins, a bunch of them have these weird coils and a bunch of them have these weird sheets. So I get alpha helices and beta sheets. Those beta sheets and alpha helices can then interact with each other, and I'll call that tertiary. So our classification of beta barrels is weird because we do see these patterns show up in other proteins. So we'll call it as a secondary structure, but it doesn't quite fit secondary, hence the secondary plus. Okay. What I think makes this even worse is that if we then push to our extremes over here, our far right, what's happened there? You have a beta barrel with alpha helices aligned along it. well, that's gotta be tertiary structure because I have beta and alpha. That can't be possibly secondary. Sorry, it's secondary plus. Okay. Why? Because that particular pattern of having a beta barrel with alpha helices shows up in a bunch of different proteins. Because it shows up in X number of proteins, someone says, okay, we gotta call that secondary, not tertiary. So that's why it kind of hovers on this secondary plus situation, because it hasn't quite made the transition all the way to officially being tertiary, and it's not very cleanly our secondary. Our secondary, for the most part, is beta sheets, alpha helices. Okay, we can get a little bit more with our beta barrel. I can kind of accept that one. This last one with our beta barrel and alpha helices, really, I got no explanation for why we would call that secondary. Okay, short of it just shows up as a common bonding pattern or a common arrangement in other proteins. Okay, so that's what the motif is really saying, that these are common arrangements. If we have those common arrangements, we call them secondary. Tertiary is characteristic for that protein. Okay? And I was done 2 minutes ago, sorry.